let's take a look at number 7 here. Fruit flies, homozygous for wild-type body and wild-type wing, was crossed with a homozygous recessive black-bodied and vestigial winged flies. The F1 offspring were then test-crossed with flies homozygous recessive for both body color and wing. And these are the results obtained for F2 generation. Okay, so now we have 2 different genes, and let's assume for now that both of these genes are autosomal, meaning they are not sex-linked, and complete dominance. Complete dominance. Assuming one allele is completely dominant for the other. So let's start by identifying the genotype of both parents. So it says that the first parent is homozygous dominant for both. We're crossing that genotype with homozygous recessive. So now if you cross these two, you can do the Punnett square, right? So the only possible gamete that you can produce from the second parent is big B, big B, big V. And the second parent is little b, little v, meaning you're going to be producing double heterozygotes as F1. Now, that's what we get from F1. In this case, to make the F2, what you are actually doing is you're crossing one of the F1 individual with a test cross subject, right? So it says test cross with flies homozygous for recessive for both body color and wing. So little b, little b, little v, little v. So this is actually how we make F2 here. If we use the FOIL method, you will know that you can produce 4 different gametes, 4 different types of gametes from the first parent. From the second parent, you can only produce one type because this parent is homozygous recessive for both. So if we complete the Punnett square, this is what we expect. So that means we expect to see 1-to-1 to 1-to-1 ratio between all 4 of these phenotypes. So let me go ahead and circle them. So we expect 25% wild type body with wild type wings. We expect another 25% wild type body with vestigial wings. We expect to see 25% chance black body with wild type wings. Oops. And finally, we expect to see another 25% chance black body vestigial wings. So that's what we expect to see from this Punnett square, right? 1-to-1 ratio. Now let's compare that to the actual numbers observed from this genetic cross. So now this ratio does not appear anywhere close to 1-to-1 ratio. Instead, we see a lot of fruit flies that are dominant for both and a lot of the fruit flies that are recessive for both. So we can do a chi-square analysis, right, by adding up the total number of fruit flies and doing the chi-square formula. However, even without doing all that chi-square analysis, it is pretty clear that our chi-square value will be humongous, right? There is a large difference between what we expect and what we actually see. So the most likely explanation is that the 2 dominant alleles, in this case, the wild type body allele and the wild type wing allele, they are linked together, meaning they are located on the same chromosome and inherited together. And also the 2 recessive alleles, the black body allele and the vestigial wing allele, they're also located on the same chromosome and inherited together. So the most likely explanation is the 2 genes are linked. Again, located on the same chromosome. The dominant alleles are inherited together. And also the recessive ones, right? The recessive alleles are inherited together. So they're grouped together that way. Okay, so that's the overview of number 7. So these two genes, the gene determining the body color and the gene determining the wing shape, they are located on the same chromosome and the two genes are linked. These two genes do not assort independently.